Hello, everyone. Welcome back to my CellRank video tutorials. In today's video tutorial, I'm going to show you how to visualize gene expression trends. So again, first we need to import the packages and the data. So let's import the package first. And we want to stop the warnings. So now we can load the data. This is the same data that we used in previous analysis. So you can see in the data, we have the planet pseudo-time, which is required for today's analysis. And also we need the magic imputed data. So we can run the planet analysis. If you want to know the details about the package of Planter, I have a video you can watch. So first we run the diffusion map, then we run the magic imputation, use the Planter package. Let's run the analysis, then have a look at the A data. Okay, you can see following the analysis in the layers, we have the magic imputed data. Because we also perform the velocity transition matrix data analysis, and we saved the analysis in the A data, so we can just reconstruct the velocity curve from the A data object. So the key will be TFW. That's where we save the transition matrix. So we can reconstruct the velocity curve. So next we can initialize the estimator. Let's run. Then we can compute and visualize the terminal states. So in order to repeat the previous analysis, we need to set the end state as 9. Then set the terminal states as alpha, beta, epsilon, and delta. And also we plotted the terminal states to confirm 4 terminal cell types. We can run this code now. So you can see now we show 4 terminal states: alpha, beta, epsilon, and delta. So finally we can compute and visualize the fit probabilities. Then the data will be ready for visualize gene expression trends. So we run the final step to prepare the data. So you can see we can see 4 images for the fit probabilities for alpha cells, beta cells, epsilon cells, and delta cells. At the next stage, embryonic 15.5, the cells are largely committed to the beta cell fate. So now the data is ready for visualizing gene expression trends. So to visualize gene expression trends, first we can have a look at the cell types and also the pseudo-time. In this data set again, we can run. You can see the NGN rate, no expression, high expression, and progenitors. Then if EV positive cells, then 4 terminal state cell types: alpha, beta, delta, and epsilon. Then here is the pseudo-time analyzed by the Planeta package. You can see from the pseudo-time image, the cells started from NGN3, no expression progenitors, gradually increased their pseudo-time to generate beta cells, alpha cells, delta cells, and epsilon cells. We can use a wiring plot to see the quantitative distribution of the pseudo-time in each cell cluster. So we can plot the wiring plot, then set the key as plant pseudotime and group the cells by their cluster name. Let's run. You can see we generated the wiring plot. The lowest pseudotime is in the NGN3 no expression progenitors, then gradually increase in the NGN3 high progenitors FEV positive cells. You can see high pseudotime in all the cells, in beta cells, alpha cells, delta cells, and epsilon cells. So at the moment, we are still looking the data. Then the first step to realize gene expression trends, we need to fit the generalized additive models to magic imputed gene expression data, and also order the cells in pseudotime, and also weight each cell's contribution to each trajectories according to their fit probabilities. We have 4 trajectories in this dataset. So to fit the GAM models, there are 2 methods you can use. You can use the PyGAM in Python and the MGCV method in R. So MGCV method is more difficult to set up. So here I'm going to use the PyGAM method. So let's run the first step to fit the generalized additive models. Now we can realize the expression trends. First, we can use the line plots. For example, we can plot the mark genes GCG glucagon for alpha cells, insulin 2 for beta cells, somatostatin for delta cells, and ENPP1 for epsilon cells. The model will be the model we just fitted above. We use the PyGAM. Data K will be the magic imputed data. That's why we run the Planter analysis when we prepare the data. And the time K is planetary pseudotime. If you performed like RNA velocity analysis or DPT pseudotime analysis, you can also analyze different pseudotime. So let's plot the gene trends for 4 mark genes. We can run. Okay, you can see we generated the figures. First is the glucagon expression. You can see the alpha cells were labeled in blue color. You can see the increase of glucagon expression in the alpha cells. And here is the insulin 2, and then you can see by the green color the increase of insulin 2 gene in beta cells. Then down here are the somatostatin expression in delta cells. Then ENPP1 increase their expression in epsilon cells. So in this plot, we have the cells. If you don't want to show the cells, you can set the hide cells as true. Then we can just see the gene expression trends without cells. We can run it, then you will see the image. You can see now we generated the gene trends image for 4 mark genes, but we don't have cells in the plot now because we set the hide cells argument as true. So I showed you how to use the line plot to see the gene expression trends, and I used the 4 mark genes. You can try other genes if you like. Then next, I will show you how to realize the gene expression trends via the heat map. So in my previous video tutorial, I showed you how to compute the lineage drivers for each cell type. So here we can use the beta cell canister as an example again to show you how to visualize the gene expression trends via the heat map. So we need to run the computer lineage drivers function again to identify the driver genes in the beta cell canister. So here we set the lineage argument as beta. After that, we can have a look at the top 30 genes in the beta cell canisters. Let's run. Okay, you can see we generated the data frame for beta cell drivers, and here are the top 30 genes. They were ranked by their beta cell correlation to the pseudotime. So we have the data frame named as beta drivers. So now we can plot the heat map for the top 30 genes. We can get the gene names from the beta drivers data frame for top 30 genes. Then the data will be A data, model will be the CAN model we fitted in the first step. Because the driver genes are for the beta cells, so we need to set the lineage here as beta cells again, canister K as canisters, then data K will be the magic imputed data, time K is the permanent pseudotime. So now we can run the heat map for the top 30 genes. You can change the number here according to your data analysis. Let's make the heat map. You can see we generated a heat map. You can see those genes down here, they have a high expression in the beta cells. And the genes up here show gradual increase along the pseudotime. Okay, this is the heat map to show the top 30 driver genes for the beta cell canister. I use the beta cell canister for demonstration. You can use other cell types to run the heat map plot. So I'm going to stop from here for today's video tutorial. In next video tutorial, we can continue our analysis to canister gene expression trends. I hope to see you in my next video tutorial.